Hi, I'm Jeff Chamberlain from the University of Washington, and I want to talk to you about another approach for gene therapy of Duchenne muscular dystrophy. In my previous lecture, we talked about methods that are designed more to restore production of dystrophin by a gene replacement approach. But another method that's being developed, which is something that may come along well into the future, is to use AAV technologies to try to correct or repair the dystrophin gene. So in other words, instead of trying to bring in a new version of the dystrophin gene, can we actually fix the mutations? And so the title of this lecture is, Can AAV be harnessed for targeted gene repair by CRISPR-Cas9? So let's pause for a minute and consider this case study. Microdystrophin AAV transfer has been shown to effectively restore the sarcolemma expression of the dystrophin glycoprotein complex, and it can inhibit myofiber degeneration in animal models of muscular dystrophy. What are going to be some of the challenges for translating microdystrophin therapy into the clinical setting? Here's some answers to that previous question. I mentioned earlier that AAV vectors are episomal vectors, and those can be rapidly lost from cells, particularly dividing cells. So could that be a problem? We don't yet know how long this method of treatment will work. The ability of AAV vectors to persist depends on the half-life of a particular cell type. If an individual is exercising a lot and they undergo damage to some of their muscles, could these AAV vectors be lost, and will the effectiveness decrease? It's a little difficult to predict the answers to these questions, but our guess is that patients may need to have their AAV treatments repeated approximately every 10 years. So let's do a 1-minute right. Think about this for a question. We've talked earlier about how we could bring a new dystrophin gene into dystrophic muscles, but could you harness this AAV vector technology to actually correct or repair a mutation in the gene leading to Duchenne muscular dystrophy? So why doesn't everybody write down their answers to that question for a minute? Well, here's an answer to that previous question, and that is to take advantage of a new system for genome editing based on the CRISPR-Cas9 system. So what is CRISPR-Cas9? This is a natural system that was identified in bacteria, and it's a method that can be used to generate targeted genetic modifications. In other words, it's a system that can go in and bind to specific sequences in the DNA of cells and cause predictable targeted modification of that DNA sequence. It can be used to target mutational hotspots. It can be used to introduce shifts within exons to restore a reading frame, for example. It can be used to delete portions of a gene, for example, to delete a mutation that's leading to a genetic disease. So what do you need to get this CRISPR-Cas9 system working? There are 3 major components of the system. One is the Cas9, which is a bacterial nuclease. This is an enzyme that can go in and clip DNA in certain places. How do you control where this nuclease is going to be active? That's determined by the second component of the system, which is known as a guide RNA. And these guide RNAs are able to identify a region in your target DNA known as a protospacer. And this is a predefined region that you can go through and search your gene of interest, find these targeting regions, and design a guide RNA that will home in and find that region and bring in the Cas9 nuclease to introduce a cleavage of the DNA at specific and predictable sites. So the way this works is that the Cas9 nuclease and the 3' end of the guide RNA form a protein RNA complex that targets the Cas9 protein to a specific region of the DNA, and then the nuclease activity in the Cas9 protein is able to clip DNA at a specific point. So how could we adapt this technology to this AAV gene delivery system that I mentioned earlier? In the previous lecture, we talked about using AAV vectors to bring in a replacement copy of the dystrophin gene or to bring in antisense oligonucleotides to skip over mutations. Neither of those approaches is a permanent method of therapy, because when the AAV vector goes away, your therapeutic vehicle will go away also. but the CRISPR-Cas9 system potentially represents a way to permanently correct the mutation within the cells. But first we would need to get it into an AAV vector, deliver it to muscle cells, and then have it act upon the mutant gene. So the way that we've been developing this approach in my laboratory and many other laboratories are pursuing very similar technologies is to try to bring in the needed components into the AAV vector system. One of the problems that we've run into, similar to the large size of the dystrophin gene, is that this Cas9 protein is quite large, and it's very difficult to fit it into an AAV vector together with the guide RNAs and the different components that you need to make this work. So the initial approach that we've taken in my laboratory is to split the different components that you need into two different AAV vectors. And I'm going to show you some examples of where we are adapting the system to target a mutation that's located in exon 53 of the mouse dystrophin gene, just as a proof of principle of whether or not we can get this method to work in muscle. So I've shown here two vectors. These are both AAV-based vectors. The top vector carries the Cas9 nuclease together with a nuclear localization sequence to make sure that it's homing into the nucleus where it has to do its work. Other components of these AAV vectors in blue are the inverted terminal repeats that are necessary for packaging an AAV DNA into an infectious capsid shell. And in light green, we have a gene regulatory cassette, which is a muscle-specific promoter and enhancer element such that this Cas9 nuclease will only be produced in muscle cells. At the bottom of the slide is the second AAV vector, which is encoding a targeting construct. In this case, we've put a couple of different things into this vector to make the experiment a little easier to follow and to analyze the results of what we've done. We've introduced two different guide RNAs, which is indicated here with the gRNA, and those guide RNAs are produced off a small gene regulatory element known as the U6 promoter. So that's active in all cells, not just in muscle cells. For convenience, we've also introduced a reporter gene, which is the mCherry reporter gene driven by the CMV promoter. That's just a genetic element we put in there so that we could follow where our vector went within different types of cells. And we've also included a template which matches the normal version of the gene sequence that we're trying to correct. And I'll come back to that in a minute to break down all these different components. So there's a couple of different strategies. We built these vectors so that we could test several different strategies at once to try to determine which, if any of them, was the most effective. The top panel here shows our area of interest. This is a close-up region, a cartoon representing part of the mouse dystrophin gene. There are a couple of introns in there and two different exons. In particular, we have exon 52 and exon 53. In the particular mouse model for Duchenne muscular dystrophy that we've been studying in our laboratory, which is known as the MDX4CV mouse, it has a single base change, a C to T conversion that creates a premature stop codon in the gene. So instead of exon 53 encoding a set of amino acids like it normally would, this mutation makes a stop codon. The translational machinery is reading along the messenger RNA that has arisen from this mutant gene. It hits this premature stop codon, it falls off. The protein that has been produced up till then is unstable, it's degraded, and it's difficult to detect by immunological methods in muscle cells. So we want to fix this mutation. And a couple of the strategies, the first one we've shown here is we designed two guide RNAs that are on either side of that mutation. We've also brought in on one of our AAV vectors a normal template which corresponds to the normal sequence in wild type or normal mice. And the idea here is that we can clip the gene on either side of the mutation and a couple of things can happen. One thing that could happen is that the normal piece of DNA that we brought in can recombine into that gene and replace the mutant template or the mutant gene with this normal template, fixing the mutation. Now, another thing that can happen, which we're testing for simultaneously, is that by clipping the gene on either side of the mutation, you then allow the natural DNA repair machinery that's present in almost every cell to come back in and try to repair this clip that you've put into the gene. And in doing so, it doesn't repair it perfectly, and you can get a very small deletion of a couple of bases being removed during the repair process. That's a process known as non-homologous end joining. And in doing so, approximately 1 out of every 3 of the repair events should have restored a normal open reading frame on the messenger RNA that comes off of this transcript, and we will have repaired the mutation. Now, in this case, we would only fix at most about 1/3 of the mutant genes, but it turns out from other things that we've seen in patients and in different animal models for muscular dystrophy, a 1/3 repair rate would have a major impact on this disorder. So we would be happy if we could achieve that. A third strategy that we've taken goes out a little further. Rather than trying to direct the Cas9 nuclease to clip right at where the mutation is, we've looked at the ability to simply clip out a larger part of the gene. So in this case, as shown here, we've designed 2 guide RNAs that will bind upstream of exon 52 or downstream of exon 53. And by clipping the gene in 2 different places, the gene should come back together, and the cellular machinery will produce a messenger RNA from this gene that simply lacks both exon 52 and exon 53. Well, what's the advantage of that? Why would we want to cut out part of this gene? Well, the reason we want to do that is that exon 53 has a mutation. It has a mutation that prevents the production of a functional dystrophin protein. If we simply removed exon 53, it has a number of amino acid coding bases in there that isn't a perfect number. So you would not restore an open reading frame to the messenger RNA. And it turns out we need to take out exons 52 and exon 53 such that when exon 51 is spliced to exon 54, you now have a messenger RNA that will make a functional protein. It has an open reading frame, but that protein is going to be missing just a little piece of the normal amino acid sequence, but that's a small enough truncation. We know from patient studies and from the mini dystrophin work we've done in the past that that type of a dystrophin will be highly functional. So any of these 3 different strategies, if we could get them to work, should lead to production of new dystrophin in muscle cells and hopefully correct the dystrophic phenotype. So let me show you some examples of how this has worked out so far in actual testing in the MDX mouse model for Duchenne muscular dystrophy. So in this experiment, we took the different AAV vectors we prepared that have components of the CRISPR and the Cas9 system in them and injected them directly into the TA, or the tibialis anterior muscle, of the mouse model with Duchenne muscular dystrophy. And what I'm showing you here are results from the 2 different approaches. The one approach, which involves the targeted correction of the mutation in exon 53, or the delta 53 as we refer to it, and the second example where we've attempted to delete both exons 52 and 53 in order to restore an open reading frame around that mutation. And so what we can see here, we're looking at a couple of different muscles that have been injected with our vectors, and we are probing these muscles in 2 different ways. The green color shows staining with an antibody that recognizes the dystrophin protein. The red color is picking up the mCherry reporter gene that I mentioned earlier in one of my slides that's in the vector, just to allow us to follow where the vector has gone within the injected muscles. So in all cases, we see a reasonably good distribution of the mCherry color, and that indicates that the vector is spreading throughout the muscle, although you may note that it's not spreading everywhere. A direct intramuscular injection approach is not as efficient as a systemic or an intravascular approach, but it's a lot easier to do, and we can get a lot of information more rapidly using this method. The important feature is to look at the green color in these injected muscles. So again, the green color is from an antibody that detects the dystrophin protein. In the absence of green color, there is not dystrophin. In the presence of green, we're seeing muscle cells that are producing the dystrophin protein. And again, the mouse model normally does not produce the dystrophin protein. So what we see in both examples here with either the delta 53 targeting strategy or the delta 52-53 deletion strategy, we are able to see very widespread amounts of the dystrophin protein in these treated muscles. You may notice that there is, in some cases, a fairly patchy distribution of the green color, so we have not achieved a uniform or 100% correction, but the exciting thing is that we are able to see dystrophin being produced in these treated muscles. Some of the muscles are making more than others. We do see variation from one animal to another. And in particular, in the example of the delta 52 to 53 strategy, where we're trying to delete two different exons, we see some larger areas where the method was not as efficient, and we're still trying to improve overall how well this approach is working. Here's another example using a different imaging method. In this case, we've used confocal microscopy to develop sharper images of the dystrophin production within the muscle cells. And again, hopefully you can see with both the delta 52 to 53 deletion approach and the delta 53 approach to target the direct mutation in the MDX mouse, we're able to see widespread regions of dystrophin production within these injected muscles. And you can compare that to an uninjected muscle. In this case, the contralateral muscle of these animals was always injected with only one of the two vectors that you need to achieve this CRISPR-Cas9 co-delivery. And in the case of the control muscle, the contralateral muscle, we don't see dystrophin production, but we are seeing significant production of new dystrophin protein in both of the treated muscles with both of the different strategies that we've been applying. So another way to look at these injected muscles is to ask, are they making dystrophin by the immunostaining approach? But how big is that dystrophin? And you can look at that by doing an Immunoblot, where you make an extract from the muscle and run it out on a polyacrylamide gel that separates different proteins by their size, and then we visualize those proteins with antibodies. So this is an Immunoblot assay. And what I've shown here are two different Immunoblots using two of the different strategies that we've been testing. One is a strategy to directly target the mutation in exon 53. That's on the right-hand side of the panel. The other is a strategy to remove both exons 52 and 53. That's shown on the left-hand panel. And the top two panels on these Immunoblots are panels where we have come in and probed with two different antibodies against dystrophin. One binds to the carboxy terminal portion of dystrophin, the very end of the protein. The other binds to the amino terminal portion of dystrophin, the very beginning of this protein. And this is a way where we can ask whether we're making the full-size intact protein or not. And as you can see, with both approaches, following from left to right, we can see the full-length protein being made in wild-type or normal muscle, and we can also see the production of a full-length protein in the treated dystrophic animals. But in the lanes under the MDX symbol, we don't see the dystrophin protein. So this is new protein that we've generated by a targeted nuclease modification of the dystrophin gene. The further down on this panel, you see the labeling SP-Cas9. That is just an antibody to detect the Cas9 nuclease, so we know it's still being produced in these muscles. And below that are antibodies against a couple of different muscle proteins just to normalize our loading, how much we put onto these blots. And the final assay that we've looked at, or one of the final assays we've looked at, is to ask if the messenger RNA that's coming off of these targeted genes now encodes what we predicted that they would encode. And the example I'm showing you here is the case where we developed vectors that were designed to delete both exons in 52 and 53 from the dystrophin gene. And if that was successful, you would predict you would have a messenger RNA arising from that gene that joined exon 51 directly to exon 54. And what you're looking at here is a DNA sequencing analysis where we sequenced the RNA coming off of the dystrophin gene, and indeed we see a perfect joining of exon 51 to 54. That the site of the joining is shown by the vertical red line at the top. and you see the G to C joining there. The G is from exon 51, the C is an exon 54, so we have achieved a precise targeted correction of the gene in this case. So just to summarize a little bit, the AAV gene delivery system can be harnessed to take advantage of the CRISPR-Cas9 gene editing machinery that was identified a couple of years ago in bacteria. We've used this approach to achieve a targeted modification and correction of the dystrophin gene in a mouse model for Duchenne muscular dystrophy. We can restore production of essentially a full-length dystrophin protein. There's still a lot of variables that we're looking at that try to improve this technology. We're looking at which gene regulatory elements are best to use to produce the Cas9 protein as well as the guide RNAs. But if this method can be continued to show great promise like this, it's a method that can lead to permanent genetic modification in adult mice and hopefully eventually in clinical applications. The amount of dystrophin we're producing is still a little on the low side, so there's a lot of modifications that are going to be needed to improve the efficiency of the method. We need to look at longer time points. There's a lot of different ways that we redesign these vectors, but what we have so far is a proof of principle that the CRISPR-Cas9 system can be applied to adult animals and lead to a targeted gene correction in adult mammals. And we'll see whether this technology can be scaled up and proved to be efficient and safe enough for perhaps going into clinical application in the future. So I will stop there. Thank you for listening, and hopefully you've learned a little bit about gene modification today.